It's preparing, so we should be on good Swiss time. Okay, good afternoon, Europe. Good morning, America, and wherever else you are in the world. Good morning or good afternoon. I'm Carsten Schradin, a researcher at the CNRS in Strasbourg, and it's a great pleasure today to host you the fine Michael Taborsky, who could be said to be something like one of the fathers of this seminar series, because when I was invited by his team in Bern to present via Zoom this year in May, it was such a nice experience for me to decide that I want to have this every week. And with the help of Lauren and Eduardo, started this seminar series here. Michael has been in other ways quite influential for my research, because when I started as a diploma student, that's something like a master student nowadays, I worked on cichlid fish, and he was then already the leading expert on cichlids. He did his PhD thesis already on cichlids, on Lamprologus brichardi, which in the meantime is Neolamprologus pulcher, but still same species. He did this PhD in Seewiesen in Germany at the Max Planck Institute in 1982, even though officially his PhD was obtained from the University of Vienna in Austria. He stayed afterwards as a team leader at Seewiesen for many years, until in 1993 he became a professor in Vienna at the CLIF, the Konrad Lorenz Institute for Vergleichende Verhaltensforschung, so institute specializing on animal behavior. And then finally in 2000, he moved to Switzerland, to the University of Bern, where he has been working since then. Michael has been a broad interest in behavioral ecology, and especially the evolution of social behavior and cooperation. And he studied for this not only cichlid fish, but a wide variety of taxa, including insects, birds. For example, they have very interesting studies on ambrosia beetles and also Schutz in Bern. They do exciting experiments about cooperation in rats. But I asked him, maybe because of my history today, to present a broad overview of his work he has been done on social cichlids from Lake Tanganyika, because I'm still very much interested in these animals. Maybe they prompt me from my diploma thesis, and I know that it's just a very exciting taxon to work on. Michael published way more than 200 papers in all journals, including Nature. He has more than 13,000 times been cited by his colleagues, had more than 40 PhD students, more than 20 postdocs. So he is really a big shot in our field, and he had a big influence on many people, including myself. And as such, it's a very great pleasure for me to introduce Michael today here to the fine, and I'm looking very much forward to your presentation.
Well, thank you very much, Carsten, for this very nice introduction. And welcome everybody. I'm really glad to be able to talk about our work on shell-brooding cichlids. It's actually not the work that we do on highly social cichlids, which Carsten already mentioned. But it is rather a project where we would like to understand evolutionary mechanisms causing adjustments, or say adaptations, to conflict both within and between the sexes. So if we think of sexual conflict, we need I apparently have problems in advancing the presentation. No, okay, now it works. Okay, so when we think of sexual conflict, we are interested in diverging fitness interests between reproductives, and this, of course, may involve reproductives of different sexes. So there may be conflicting fitness interests between males and females. For example, regarding the reproductive investment and the use of important resources. But there's, of course, also intrasexual conflict, which means there's competition for mates and fertilizations. What I would like to focus on today is the ecological dimension of sexual conflict, and in particular on the effect of breeding substrate on sexual conflict and on the selection of adaptive strategies both in males and females. Males provision females with breeding substrate very often. That is a very general pattern through many taxa. And females use these substrates then for optimizing their reproductive success. One example is the so-called pendulant tit, a European passerine, where the males build very complex nests and the females choose males independent of the size of the initial nest construction. So the females already decide at the point when the nest is far from being completed whether she will prefer this particular male or rather search for an alternative partner. And in the end, these very complex nests determine female success, because the larger the nest, the more insulation, the higher the reproductive success of females. So they can really judge the quality of a nest already at an early stage and select males independent of that. So that shows that the breeding substrate can be important for intersexual relationships and also for potential intersexual conflict. In the aquatic environment, mollusk shells are a very frequently used breeding substrate. If we think of aquatic invertebrates, for example, hermit crabs, this is a hermit crab with its a big clutch of eggs inside of a snail shell, or of octopuses that in many species use bivalve shells to protect their brood, then it is clear that mollusk shells are an important potential resource that serves as a breeding substrate. Sometimes octopuses end up in not so typical shells, which are different from mollusk, of course. In Lake Tanganyika, where we find many cichlids that what we call substrate brooding. That means that they are not mouth brooding, which is the most common form of brood care in cichlids. But in Lake Tanganyika, there are many species that raise their offspring on the ground or in shelters. And one of these potential shelters are mollusk shells. And there are even 11 obligatory shell brooders, which means that they only breed in shells, like this species here, Lampologus ocellatus, that buries shells and raises the offspring. Here you see two young emerging from a buried shell within this protective substrate. There are many more species of cichlids in Lake Tanganyika that use shells, but these 11 species only use shells. And what you can see in this picture below now is what we speak of as a shell bed, which means hundreds and hundreds of thousands of shells packed together, which may be very old. In some parts, it has been shown that these shell beds exist over tens of thousands of years, which is due to the fact that in Lake Tanganyika, the water chemistry is very basic, which means that the shells do not deteriorate. They are not dissolved by acidic water. And by secondary calcification, they can really build reefs, like in this picture shown below. So this is really an important substrate in Lake Tanganyika for many cichlid species to raise offspring. If we return to our main question, the conflict within and between the sexes, there is an intriguing example among these shell breeders in Lake Tanganyika, and this is Lampologus calypterus. Lampologus calypterus is a species that breeds in snail shells, like the ones that you see here below. And that shows an enormous sexual dimorphism. This is a male, nest male that defends these shells as a nest. And this is a female here caught in or shot in the field. And you see the size difference between them. That is quite typical. In the lab, where you can see that perhaps a bit better, this would be male and female, just to show you the relationship in size and coloration. So in nature, the territorial males collect shells, either big shells like in the top graph or in the top picture, or smaller shells if only small species are available in the lower picture. And they build nests to attract females, and the females then enter these shells for spawning. These shells provide, of course, magnificent protection for raising offspring, for the eggs and for the larvae inside. And the female leaves these shells only after 2 weeks of caring after depositing the eggs. The males, the nest males, are much too big to enter these shells. So you can see here one of the shells where a female is lying inside, that is raising offspring, just caring and guarding the eggs and larvae. And the male is sniffing into the shell entrance to get information about the status of the female and the offspring in the shell. The males, of course, protect the shells against predators and also against conspecifics. We will get into that in a moment. This species, Lampologus calyptrus, shows actually the greatest sexual dimorphism among animals, with males exceeding females in size. In the field, this sexual size dimorphism is 1 to 12. So males are 12 times heavier than females. If you think of enormous sexual size dimorphisms in the direction of large males, small females, you may think of elephant seals, for example, which are or have been known before Lampologus calyptrus turned up as the world record holder as an extreme case, but there the ratio is only 1 to 7. So calyptrus is really exceptional in that direction. What is the reason? I mean, what could be the reason for such enormous sexual dimorphisms? Obviously, this may result from intrasexual selection, from male-male competition, or from a female choice, which is a measure of intersexual selection. It may also be that there are ecological constraints on male size, or there may be ecological constraints on female size. So for the next few minutes, I will address these different possibilities. And first I will report on an experiment that we did a long time ago in Vienna in a large circular tank. This is work that was done by my PhD student and later postdoc, Dolores Schutz, where we tested these different possibilities, male-male competition, in this setup here, in this part of the tank. I hope you can see the arrow of my pointer, where we had large and a small male together with 20 snail shells and 4 females, and the females could choose between these different males. In another section of the tank, we tested for female choice. So the females could choose between a small and a large male, and they could move through a perforated wall where the males could not pass. And we measured whether they would prefer to spawn with a large or the small male. And then we had a section where we tested for the limitation of males in body size. So we provided the males with shells for a nest, and then we provided single shells of different sizes to the males at a distance of roughly 2 meters, where they had to collect the shells to enlarge their nests. So, as I said, it was a combination of male-male competition, female choice, and ecological constraints test on body size. And what we found, I do not show you all the data because I have much more things to present. But these are the results both from fieldwork and from this lab experiment. Large males do not hold larger nests, and large males do not outcompete smaller females. So there's very little evidence for intrasexual selection to be important for this size dimorphism. Regarding intersexual selection, females did not, in our experiment, did not prefer larger males. And that is also supported by field data. But the females were attracted by larger nests. So here in this graph, you see the number of snail shells per nest. You see that some nests can really have hundreds of shells. The white circles denote the available shells, the total number of shells in a nest, and the diamonds denote, the black diamonds denote the number of shells that are really suitable, that are big enough and that are in a good condition, that are not broken to females. And you see that there is a very strong relationship between the female number that will additionally end up in such a nest for spawning and the available snail shells in such a nest. So what is the ecological constraint on male size, or is there any? Yes, there is, and that is the carrying capacity. And this, I mean really literally, males only can carry shells if they pass a certain body size. So here on the x-axis you have male size, standard length, which is the size between snout length and the beginning of the tail fin. And on the y-axis you have the number of transported shells in that experiment that I showed you. And you see that below 9 centimeters, males were not able really to collect these shells, but above 9 centimeters apparently they were. And importantly, the larger the male, the higher they could also carry the shells. And that is important for larger distances. If they collect shells meters away, we found in the field they can carry shells for 7-8 meters, then they have to reach a certain height in the water column to be able to carry the shell for such a long distance. So they are also more efficient the larger they are. And the reason for that is actually that these shells are extremely heavy. They are really strongly calcified, and therefore the males need the power which they only get by a certain body size to be able to build these nests. Well, are there also ecological constraints on female size? If we look into the choice of females in the field, we find that only the largest shells that are available in Lampologus calyptrus nests are occupied. So the gray distribution here shows the shells that we found in numerous nests in the field. The black columns show the shells that are occupied by calyptrus females, and the striped gray, dark gray columns show the occupation by other species within calyptrus nests. So other species also use these collected shells for breeding. But what is really remarkable that only the sort of the upper end of the size distribution of shells within these nests of males, of bourgeois males, are used by the females. And even within nests, if we look into which shells are taken first, so this is the biggest shell here on the x-axis, the second biggest, third biggest, and so forth, you see that the number of females that first chooses the biggest shell is highest. So they really select large shells. And the reason for that is that, and here you see the preference of females both in the field data set, the dark dots, and elaborate data set, the gray diamonds. And you see that in both cases there is strong selection for shell size independence of female size. So small females apparently select a shell that is fitting their own size. They're not going for the biggest shell. And if you think of, if you ponder about why females select large shells in the first place, the reason is that they produce a larger number of young in a large shell. So this is the number of young that survived in these shells, independence of the residuals of the relationship between female size and shell size. So the larger the shell in comparison to female size, the more young they can successfully breed. So apparently, males and females benefit from large size. Larger males because they can have larger harems, so they have larger nests. They are able to carry shells more efficiently, and that means that more females end up there. And the females, of course, benefit from what we call fecundity selection, because the larger females can, of course, produce larger clutches and raise more offspring. They need larger shells, of course, and there is a limitation, as we have seen, in the shell size distribution within these nests. But anyway, the larger the female overall, the more its success. So we thought about how to model male and female size to really find the responsible selection forces. And together with Jeff Parker, we tried different modeling approaches using a life history model and the gain rate maximization model, or combination of both. We ended up predicting female size quite nicely within the range of sizes that we find in the field. So these are the field data with median and interquartile ranges and range. And that fits quite well to the predicted values, both of the gain rate maximization model and the combination with the life history model. Our prediction of male size, of nest male size, from the life history model did not match the field size distribution of nest males. It was much, much larger. So the smallest males, this is the range, as I said, were roughly 10 centimeters in size, even though we predicted with our model a size of, say, 8 centimeters where they would start reproduction. And the reason for that is really the ability to carry shells, which of course was not incorporated in this model. So it shows that really the males are limited by this ecological factor that apparently is responsible for the large male size. However, we have here another male type. That is what we call sneakers. Sneakers are reproductive parasites that are opportunistic and trying to fertilize eggs while a nest male is spawning. And they are apparently in size. These again are field data. And this is the size distribution of sneakers in the field. They are in size between females and nest males. So these males are opportunistically visiting nests and trying to fertilize eggs from the shell entrance when a female is spawning inside these shells, and when the nest male is inattentive, for example, because it courts another female or it defends the nest against intruders. Interestingly, we have another type of male in this species, and that is what we call dwarf male, for good reasons. If you look at the size difference between the nest male and the dwarf male, then this is enormous. The nest males are 60 times the mass of dwarf males. And the special role of dwarf males is that they enter the shell, they pass a female that is spawning in the shell, they enter into the tip of the shell where they fertilize the eggs from a very privileged position. They're very close to the clutch that is released by the female. So this will be a spawning nest male releasing sperm in the field, and this will be a view behind closed curtains. You see that inside the shell there is a female and the dwarf male that has passed the female and now fertilizes the eggs from inside the shell. So these are the different sizes. Nest males are between 11 and 13.5 centimeters standard length in the field. Sneakers are slightly smaller. Of course, there are immature males also that are not yet reproductively active. But the dwarf males you see here, they are really, really tiny. No size overlap with these other male types. So we tried to find out how an individual male decides whether it becomes a nest male or a dwarf male. And we use different experimental approaches. For example, in one experiment, we raised offspring of nest males and dwarf males under standard conditions and collected data over a long period to measure their growth trajectories. In another experiment, where we tested for potential environmental effects on growth, we varied temperature, we varied the number of shells, and we varied the available food in offspring from nest males. And now it was both male and female offspring, daughters and sons, and looked into the effect of these environmental variations on growth trajectories. And what we found, shortly summarized, was very remarkable. We found, and here is, you see the standard length against age. And in the left graph, you see the growth trajectories of male offspring, in the right graph of female offspring. And what we found, a very divergent pattern of males that were sired by nest males and by dwarf males. So actually, the dwarf male sons grew quicker, and that was a significant difference. They grew quicker in the beginning, but very soon they leveled off. Already after about 100 days or so, they grew more slowly than the nest males, and they stayed at that size for years. Whereas the nest males continued growing. I mean, there are bony fish which do not really stop growing usually, except these dwarf males. In females, the father did not make any difference. So dwarf male daughters and nest male daughters did not grow differently quickly. Of course, there were effects also on growth patterns, on growth speed by our environmental variation, but it did not influence whatsoever the divergence between nest male sons and dwarf male sons. So that by a standardized lab experiment showed in the pedigree analysis that really only the father of the offspring determined the growth speed. So these are various pedigrees, and I just would like to explain one very simple type. So if this is a nest male, the blue one, and the red one is a dwarf male, then the son will be the dwarf male type and show this growth pattern if the father was a dwarf male, irrespective of the father of the grandfather. And this is the opposite case down here on the right side, just that you see that really only the father determines the growth pattern of the offspring, which led us to think that this is really a sort of very simple Mendelian Y-linked genetic polymorphism. And when looking at the genetic data, we found a 4.2 megabase per sex region with tightly linked candidate genes for both body size and sexual development. And what is most interesting in that analysis is that we found allelic variation of candidate dwarf genes that distinguish dwarf males from giant males. And this is actually a gene that is known from pituitary dwarfism in humans also. So apparently this seems to be a very conserved genetic trait. The gene is apparently under strong purifying selection in the Lampologus calyptrus trait, shown by the DN-DS ratio of nearly 0.07. And interestingly, also behavior-associated genes are located in the same sex region. So our analysis revealed, and this is a study done by Puya Singh, who is supervised by Christian Sturmbauer and myself for her PhD. So what we found, or what she found, was that there is a very high male-female FSD, which suggests opposing selection due to sexual antagonism. And interestingly, the evolutionary strata that we propose show a strong annotation of sex-determining and body size genes. So this leads me to switch to a sort of more general theme, which regards the evolution and the evolutionary stability of alternative reproductive tactics. And we are now focusing on alternative reproductive tactics in the male sex. So in males, generally, when you find alternative reproductive tactics, you find bourgeois types of males that invest in monopolizing resources and mates, and often also show brood care, not necessarily, but often they also show brood care in many different taxa, not only in fish. And then you have parasitic males that exploit the investment of these bourgeois males by scrounging fertilization attempts. And this has been shown, for example, and well studied in ruff. This is a very important bird example that has been well studied, or also in many fish species, including salmon. And a key feature of these alternative reproductive tactics is, first, that you have a variation among individuals. Second, that you have more or less consistency within individuals. So you can either have flexible tactics that respond to the current situation, like the opportunistic sneaking tactic that I showed you in Lampologus calyptrus, or you can have fixed tactics. And fixed tactic can come about either by a developmental switch or a threshold. And when this threshold is passed, then the individual has decided to go into one or the other direction regarding its life history trajectory. Or you can have inheritance of alleles of lag effect. So this is completely unconditional and solely triggered by the genetic underlying this dimorphism. And the third feature that is important is that you find correlations across contexts. And that not only includes reproductive, but also non-reproductive contexts like the growth trajectories or feeding regimes and the ways how animals deal with their ecology. So as I just outlined, alternative reproductive tactics may be fixed for life or flexible. And this is a simple graphical scheme, how you can imagine how it works. You have a juvenile phase, which is this striped part of this horizontal column. And that develops either in a bourgeois type, the white column, or in the parasitic type, the black one. So this would be fixed for life. And in the plastic case, you can either have a simultaneous change between these two different tactics. So throughout life, you can always decide, dependent on the current conditions, whether you are better defending a territory and courting females, or whether you use the effort of other males by parasitizing the effort with a sneaking tactic. And you can have a sequential tactic choice. First, when you are small, using the parasitic tactic, and then switching to the bourgeois tactic when you are larger and more powerful in defending resources. Or you can also have reversible sequences. All these different cases are known from animals, especially from many different fish species. Now we go back to our example, to Lampologus calypterus. And there we looked into the consistency of behavioral differences across different contexts between territorial males and dwarf males. And what we see in this graph is the percent of time that they spend with different activities. And let's focus on feeding, courtship, and aggression. And you see that there is a remarkable difference between these two different male types. So if we look into courtship and aggression, the territorial males spend quite considerable time in these activities. But dwarf males don't. They don't court at all, and they show very, very little aggression. Whereas dwarf males spend a lot of time during their reproductive period in feeding, and territorial males do not feed. They do not feed at all while they are defending a nest. So that shows that the difference in size and in the reproductive tactic also extends to remarkable and important behavioral differences. If we look into body condition, we find that there is also a very important difference between nest males and dwarf males. If we just focus on nest male and dwarf males, we see that nest males have quite a bit of body fat in comparison to dwarf males. Dwarf males have very, very little fat storage. Sneaker males have even more fat than nest males, but the reason for this difference is that they feed to get a large reserves for the period when they then defend the nest. When they start, sneakers are at the stage before starting to defend the nest. And when they defend the nest, as I just said, they don't feed for on average 1 month. The average nest holding period is 33 days in the field. And during that period, the nest males completely rely on their reserves. And this graph shows that they reduce their fat reserves and their weight, of course, also, but this is body fat, with increasing time of their nest holding period. So apparently nest males are capital breeders because they accumulate reserves that they can use then during the reproductive period, whereas dwarf males are what we call income breeders. They just feed constantly and use the energy immediately for producing sperm and for their reproductive activities. Now let's look at the gametic level, how dwarf males do in comparison to nest males. And we found when analyzing the performance of sperm, we found interestingly that dwarf males have very potent, very powerful sperm right after releasing the sperm. So during the first minute, they are much more effective in swimming linearly towards the egg than nest male sperm, which you can see in the comparison between the blue line, which is for dwarf males, and the red line, which is for the nest males. But this comes at a cost. The dwarf male sperm apparently are exhausted much quicker than nest male sperm. So their sperm performance declines much more quickly, and they are also not as long-lived. Nest males have much more long-lived sperm, which makes a lot of sense if you think of where the sperm are released in comparison between these two different male types. Okay, now let's focus on the coexistence of alternative reproductive tactics within a species. Of course, this is an important question. How, I mean, you may think one of the two strategies is perhaps more successful. How can you stabilize a certain relationship, a certain relative number of both male types in a population? So if we again think of the two possibilities, that either you have conditional or plastic alternative reproductive tactics or fixed tactics. In the conditional case, the two different tactics do not need to produce similar lifetime reproductive success. If you think of a variation of fitness, of potential reproductive success with size, then often the parasitic tactic is more successful with small size, yeah, because the males of small size are less conspicuous and they may be easier participating in reproduction parasitically. But after passing a certain size threshold, the bourgeois tactic is much better because these males are now more powerful to defend resources and to attract females. But importantly, in this case, when it's a conditional tactic, these two different tactics do not need to render similar reproductive success over a lifetime. And this is different if you have fixed alternative tactics, because in this case, if you have here the proportion of bourgeois males in a population, if there are too many bourgeois males so that they have a lot of competition among them, then the parasitic tactic would be more successful. Yeah, you have fitness on the y-axis, and that would drive the population towards the equilibrium state in this particular graphical case of 50-50 bourgeois against parasitic males. So we can predict that when organisms change between tactics according to conditions, so we have a flexible, plastic or conditional tactic, the mean lifetime fitness between individuals using one or the other tactic may differ. However, when the tactics are fixed for life, then the mean lifetime fitness of individuals using arts should be equal. Now, do the two male types render similar lifetime reproductive success that we find in Lampologus calypterus, the dwarf male type and the bourgeois male type? What do we need to know to compare the lifetime reproductive success of these males? We need to know the survival probability to reach reproductive age or reproductive status. We need to know the time that they can reproduce once they have reached this reproductive status, for example, the number of days. And we need to know the reproductive rate, for example, the number of spawnings, in our case, in a certain time period. And the product of these three factors then provides us with information about lifetime reproductive success of these two different tactics. And we calculated this with data that we collected in the field, and according to the behavioral data, we find that dwarf male spawnings per lifetime are quite similar, not exactly, but quite similar to the spawnings of nest males during a lifetime. Whereas if we compare nest males with sneaker males here in the right-hand graph, we see that sneaker males, you can hardly see that this is above zero, have almost no reproductive success over lifetime in comparison to when they perform as a nest male. So there is a huge difference in this conditional tactic in the relative success rates in comparison to the fixed rates, which accords with our predictions. The reason for this similarity in reproductive success between nest males and dwarf males is actually that the dwarf males that participate in fertilization of clutches are extremely successful. So if they are able to enter a shell where a female is spawning, then on average, they fertilize 3/4 or slightly more even of the eggs in the shell. So this shows the paternity of dwarf males, the light gray parts of the columns, in comparison to nest males, is the dark gray, or other males, which are usually sneakers, the black parts of the columns, in 10 different nests. And on the y-axis, you just see the clutch sizes in these different nests. So if we compare the male lifetime reproductive success between two alternative estimation procedures, either the behavioral one that I showed you already, or the genetic one based on our genetic data, microsatellite analysis of siring success in the field, then we see that these are the behavioral data on the left side that you have seen already, but also with the paternity data, that the lifetime reproductive success of the two tactics are really similar, within the same range at least. And we have seen that the sneaking tactic is much less successful. Okay, so the conclusion is that fixed tactics, and this corresponds in our case to the nest males and dwarf males, have a rather similar lifetime reproductive success, whereas the parasitic sneaking tactic is much less successful than the other tactic, which confirms the predictions of former models of frequency-dependent effects. But what about the relative frequencies of male genotypes in a natural population? And that is quite surprising, actually. The relative frequency of dwarf males is only roughly about 5% in the field. In the breeding colonies of nest males, there is about 15% dwarf males. The success of entering a shell is around that, also 14%. The broods with dwarf male paternity, where dwarf males are at all successful, is only about 5%. However, their fertilization success, if they are able to enter a shell during spawning, is actually more than 3/4, as I said already. So we may ask, why are dwarf males so rare? And one potential answer is the role of females. Females may avoid shells with dwarf males, and they may block the shell entrance against entering dwarf males. These are two observations that we made in the field. And to just find experimental proof for the fact that there is negative frequency dependence in this species, which would predict that with a high ratio of nest males to dwarf males do better and opposite. We did an experiment in the large circular tank where we varied both the relative and absolute numbers of nest males and dwarf males. So this would be 10 nest males, 2 dwarf males, 20 nest males, 2 dwarf males, and so forth. In each of these experimental treatments, we provided them with 120 shells for breeding and 20 females. And in total, during these experiments, 35 broods were produced. And we collected, of course, behavioral data and paternity data. The prediction was that with a lower dwarf male frequency, the dwarf males should have higher success, and vice versa for the nest males. And these are some of the data that we collected there. They are still being analyzed, but just a few data that I would like to show you to understand both the behavior of males and females in these different conditions. So this is the time delay until spawning commenced, until females started spawning, with a low dwarf male density or a high dwarf male density. And you see that there is a significant difference. When there are few dwarf males, the females start earlier to breed. So apparently they're less critical about which shell to enter. And not only the latency until breeding or spawning starts is different, but also the duration of spawning. With a low dwarf male density, females spawned longer, which means they released more eggs because they release one egg at a time every about 2.5 minutes. So the time, the duration of spawning also is a good measure of the actual clutch size. So few dwarf males, the females produce more eggs. If we look at the nest male behavior, at low dwarf male densities, the latency of a nest male to attack a dwarf male was shorter. So they attack dwarf males earlier when there are few dwarf males around, which was quite a surprise for us. And if you consider that in this tank also there were many males that played the sneaker tactic, aggression against sneakers was also quicker and higher in the cases where there were fewer dwarf males, so in the 10:2 and 20:2 setups. So apparently the nest males can respond more efficiently against dwarf males and against sneaker males when there are few dwarf males around than when there are many dwarf males around, which points towards a sort of limitation of the nest males when there are too many dwarf males. At low dwarf male densities, the sneaker intrusions were also shorter because of the high attack rates of nest males. So the nest males were apparently also more efficient in defending the nest against sneaker intrusions in low dwarf male densities. So all these results suggest nest males did better at low dwarf male densities, which is of course opposite to our prediction. And therefore we should look at the male success, the relative dwarf male success in fertilizing eggs in these different relative and absolute densities. Dwarf males were only successful in 4 of 35 broods. So this is only in 11% of the produced broods. Their fertilization rate varied greatly, like in the field, as we have seen, between 13 and nearly 100%. But paternity was equally distributed between treatments. Of course, with only a sample size of 4 broods where the dwarf males actively participated, there is very little resolution in this data set. So we have to conclude that there is no evidence for negative frequency dependence from our experiment. Of course, we have this evidence from the field, but not from this lab experiment. So to get back to our questions why dwarf males are so rare, we should think about who really competes with whom in this race for fertilizations between different male types. So we did an experiment, and this was an experiment done by Corinna von Kürti, where she exposed a nest male with females and shells to either a nest male intruder, to sneakers, to dwarfs, or to a mixture of sneakers and dwarfs. And just to cut it short, the test showed that males were mainly aggressive against nest male competitors, so against the same tactic, and much less so against sneakers and the mix of sneakers, and especially also against dwarf males. So that fits very well to the predictions of frequency-dependent selection, that the major competition is within your own tactic. Dwarfs, I do not show this data, dwarfs hardly attack each other, so they seem to scramble. They rather benefit from being fast than from being aggressive against other dwarf males. If we look into post-fertilization sexual selection, where the females, of course, also have some input, and the males, especially the nest males, have the possibility to sort of modify the relative success of offspring that were fertilized by themselves or fertilized by others. We found that in the field, when a male is exposed to a shell where another male fertilized all or most of the eggs, they try to expel the females from these nests, which of course would also kill their offspring. So this is a field shot with a male defending a nest and the camera where we observed the males for their entire reproductive cycle. And by finding that the males are sort of expelling females with eggs that were fertilized, or with offspring that were fertilized by other males, we did a field experiment where we took shells with offspring that were sired by other nest males in a different nest and deposited in our focal nest, where we put our camera to observe what happens. So these were these experimental shells. And on the y-axis, you see here in the upper part of the graph, the exploration parts, which means that the nest male explores the shell. He puts his head inside and sniffs into the shell. And this will be the manipulation parts, where the male tries to expel the females by taking up the shell and turning it around. And they are quite successful with that. It usually takes between 1 and 1.5 days until they can expel the female by this tactic. This will be other shells. These are shells that are unmanipulated in the same nest. These will be control shells that were also taken from other nests, but without females inside, or without females that had spawned. And this would be other shells again in the same nest. And we see that there is no difference in this control situation, but there is a significant difference in shell exploration and in shell manipulation between shells that contained offspring that were not sired by a nest owner. And interestingly, this only happens, or it affects nest male or also female reproductive success, when we do these manipulations during the early larval stage. So this, on the left side here of this graph, is the egg stage. And we see that there is no significant difference in the proportion of successful broods, which means that they were raised until the end of the larval period, between the experimental manipulated shells that contained foreign offspring and the two different control situations. However, there was a huge difference in the proportion of successful broods when we did the same manipulation during the early larval stage. There was no difference again during the late larval stage, but shortly after the eggs have hatched, apparently the nest males are able to sense by their exploration bouts that we have seen in the upper graph to sense that there are offspring inside the shell that they have not sired. So this leads us to do a lab experiment, and this was an experiment done by a master student of mine, Jonas Leuenberger, where he had a nest male with a shell and a female inside and the clutch, but he released the odor from a tank with larval cues. And these larval cues were either from a brood that was sired by the nest male or sired by a different male. And what we found was actually quite nicely confirming our results from this field experiment. Only when the odor from early own larvae was compared to early foreign larvae, there was a highly significant difference between the number of shell manipulations. Only when they sensed early foreign larvae, early means that they have hatched relatively recently, they were able to sense that these are foreign offspring, and then they started to manipulate the shell to expel the female. Not in the egg stage and not in the old larval stage. And then Jonas manipulated the relative and absolute frequencies of own and foreign larvae in this container where the larval cues were collected. And we found, very much to our surprise, that it is actually the number, the absolute number of foreign larvae that makes the difference in male responses. It is not the number of own larvae, which seems to be more sensible. I mean, it would make more sense for them to care about how many own larvae they have, but apparently they do not take effect of that number, but only to the number of foreign eggs. Not eggs, sorry, foreign early larvae. So now at the end, I just would like to focus on the female perspective of this interaction between males and females. And we should first think about the problem that females face by being in a shell with a high proportion of dwarf male offspring, because then, as we have seen, the nest male may expel her when the larvae are hatched, and that would mean that the entire brood is lost. So how do females prevent brood loss? Well, first they can choose a competent nest owner, and this is what we have seen already, this graph. so that females apparently choose males that have many shells in their nest, which means that they are a competent nest male that can not only collect but also defend shells very successfully. Sorry. And they can also select large shells, which, as we have seen already in the early phase of this talk, would mean that they can successfully raise more offspring, because the ratio between female size and the volume of the shell is important for their success. So we did a field experiment where we exposed both females and males with different sizes of shells to look into choice. The males to collect the shells, of course, for their nest, and the females to enter the shells for spawning. And we found that females very clearly prefer big shells. We had old and new big shells against small, old and new small shells, and we had control shells, and we see that which are too small actually to enter. Of course, the females cannot choose these ones, but interestingly, the males show a different choice pattern, the nest males, to the females. So that led us to hypothesize that the males may actually collect shells, even unusable shells, as a kind of extended phenotype to attract females, because it shows that they are competent defenders of a territory. The second possibility how females can prevent brood loss is to avoid spawning with dwarf males, because if the proportion of dwarf male young is too large in a shell, then we have seen the nest male will punish the female after hatching of the larvae, and so the female loses the brood. So do they really avoid spawning with dwarf males? Well, it may not always be such a good strategy to avoid spawning with dwarf males, as we can see in this graph. This is on the x-axis, you see the time development from start of spawning. Spawnings last on average 8 hours. And we see that after 5 hours, the nest males run out of sperm. This is the sperm number per ejaculation in the course of the spawning. And we see that in the later stages of spawning, the nest males may really run out of sperm. They're apparently really sperm limited. And this is from data where they spawned only with 1 female at a time. In the field, they sometimes spawn with up to 4 females at a time. So it may be that the females do well to accept dwarf males in the later stages of spawning. Let's see whether this is the case. I mean, we calculated when a female should actually allow a dwarf male to enter. And these are the eggs sired by nest males over time. And we see that this levels off because of sperm limitation. And this is this would be the number of eggs that could be fertilized if the female lets the dwarf male in, for example, after 320 minutes, so after more than 6 hours. And this would be after more than 410 minutes. And we see that the female can gain quite a bit, and this will be actually the optimal time when the female should let in the dwarf male, because it means that the dwarf male does not sire too many offspring, like here in the left part of the graph, which would mean that the female risks to lose the entire clutch because the nest male will expel her offspring. So what do females let dwarf males in irrespectively of when a dwarf male tries to get into the shell? They actually let dwarf males in within 15 minutes of the dwarf males trying. If the dwarf male is not successful, then they will leave. But what does this mean? That the dwarf male is actually blocking the shell entrance for a period of up to a quarter hour. We measured the oxygen content in these shells just to see whether the dwarf male can sort of force its way into the shell when the female is blocking the entrance. The females regularly block the entrance and do not let dwarf males in. The dwarf male is trying to get into, and in that trying, it is blocking the shell entrance. And we see here the difference between a female with a dwarf male dummy. We didn't use live dwarf males, but a dummy. that was playing the role of the dwarf male, we see that there is a highly significant difference between an empty shell and a shell containing a female with regard to the reduction of dissolved oxygen that would be available both for the female for breeding, but also, of course, for the eggs that she has already deposited. So it seems as if the dwarf males can really sort of enforce their way into the shell. So if we now summarize, what are the trade-offs that males and females of this species face? The females face the trade-off between maximizing fertilizations or brood survival. Yeah, maximizing fertilization would mean that they let in dwarf males after a certain time period. Maximizing brood survival, they should not let in dwarf males, at least not too early, because of the punishment of nest males, as they would not allow the female to raise foreign offspring in their nest. And another trade-off that females face is to submit either to dwarf male pressure, which is caused by this oxygen depletion that I just showed, or to nest male pressure by the punishment if a dwarf male has sired too many offspring in her shell. The nest male perspective also includes some interesting trade-offs. One is to either minimize stolen fertilizations by defending the shells very well against especially dwarf males, but also sneakers, or to later, after fertilization, eliminate foreign offspring. So not care too much about how many offspring will be sired by other males, but rather punish the female if too many offspring have been produced by other males. So they can invest in early reproduction or in early nest holding capacity also when we compare the sneaker tactic with the nest male tactic. Early reproduction would mean that they do not invest a lot in growth, but rather in reproduction early in life, which would hold up or delay the potential for defending an own nest and starting their career as a nest male, so to speak. Which trade-offs do the dwarf males face? Well, one trade-off is to minimize body size to facilitate shell intrusions, or alternatively, to grow big to boost sperm reserves. Of course, the tiny little body size of dwarf males limits their ability to produce many sperm. And of course, in this sperm competition situation, this is a disadvantage. And the second trade-off for dwarf males is to either attempt shell intrusions, the role that I explained to you, that they pass the female and fertilize eggs from the tip of the shell, or to try to fertilize eggs from outside, which they actually also do if there is a chance, like sneaker males do. Just release eggs from outside. So if they get too big to be able to enter shells, especially if a large female is in a small shell, they can still try to fertilize single eggs from outside the shell. And if we just, because time has proceeded so far, focus on the lower graph here. Here we have the female body weight to shell volume ratio. So to the right, we have large females with small shells. And we see here male body weight of dwarf males, and this is a field data set. And we see the squares are successful males, where dwarf males were able to enter the shells, as against failures, which are the black triangles. And we see that all the successful points are in the left lower quarter here, which means small dwarf males, small body size, and a relatively low female body size shell value ratio. That really shows that dwarf males are limited by their own body size, and especially also in combination with the size of the female compared to the size of the shell. Okay, so let me draw general conclusions now. Nest males and dwarf males are apparently blackmail females in opposite directions. Dwarf males try to force their way into the shell, even if the female is reluctant, and nest males can punish them if they have let the dwarf male in too early and too many offspring are sired by the dwarf male. The female response is apparently determined the relative frequency of male types produced, yeah, because they are really deciding how successful dwarf males are. And what I think is the most important, probably the most important message I would like to convey is that the breeding substrate may be responsible for disruptive selection, rendering extreme sexual dimorphisms, both intrasexually and intersexually, as I think can be nicely demonstrated in Lampologus characteris. So I think we should, when we're interested in sexual conflict, we should not forget the ecological dimension when trying to understand the evolutionary reasons for adaptations to this sexual conflict. Well, these studies, of course, were done by many collaborators of mine, including mainly Dolores Schutz and Corinna von Kürti, whom I mentioned already, Sabine Wirzukania, and many others. Also, theoreticians like Jeff Parker, who helped us to model the situation. And I would like to thank you for your attention. I'm sorry that it was a bit long, but I had a lot of material that I wanted to present. Thank you.
Great, Michael. Thank you very much for overview of really, I think, more 2 decades or more decades of work, quite fascinating. So we start the questions and answers now. If anybody has a question, then simply write question mark into the group chat, and I will see that you do have a question. I hope to be able to keep track of this. So if you don't type too many other comments in there to everyone, it will make it easier for me. I can start with a question that might come a bit unexpected, but you mentioned this, and it's because we got today a paper accepted in Journal of Mammalogy of nearly indeterminate growth in African striped mice, which for mammalogists is something very unusual. And you mentioned that these bourgeois males, like is typical for bony fish, they kind of grow indeterminately. They grow throughout their entire life, while dwarf males do not. But if I saw your figure correctly, the females also stop growing at some stage. Is this unusual for fish? Might this also be a specific adaptation in that species, which we won't find in other species of fish?
Yeah, that is a very good point, because of course females are also limited in their body size at the upper age. Yeah, so if they get too big, they don't find any shells anymore. And we did an experiment. Thank you very much, Carsten, for this question, because I can explain this also. We did an experiment where we raised females from 2 different populations, from a northern populations where females are much smaller and where the shells that are used belong to different species, that species of snails that produce much smaller shells, and the southern population with large shells and where the females are also larger. And we raised them under different conditions, like in the experiment that I mentioned, where we varied the environmental conditions to measure growth trajectories. And we found that, that the females from the northern populations also remained smaller in the lab situation in the F1 as compared to the females from the southern population, but the shell size that they ended up in, no, I mean the shell size that we provided to them determined their final size. So if we kept small females from the northern population with large shells, they grew actually larger than the females that were kept in a similar controlled experimental situation with small shells. So there is apparently both. There is a genetic component to female size in this species, but there is also, in addition, an environmental component. And that shows really the importance of the breeding substrate also in limiting the growth capacity of females.
Okay, that's really fascinating. So now the first question comes as predicted or said by Cheyenne, please.
Hi, great talk. I just was wondering, when you were talking about how the dwarf males are attacked earlier when there's like lower densities of them, I wondered if the dwarf males, since they're not really as aggressive, if they ever kind of like go together into the nesting males' territories.
No, I mean, this is an obvious question, like because actually the sneaker males, they go together. This is what we call a group rate. They really try to fertilize eggs if a female is spawning by entering the nests in a group. Usually, I mean, sometimes they're also alone, but usually it's 2, 3, or even up to 10 males at the same time, which of course confuses the nest male, and they more easily can participate in reproduction then. But the dwarf males are in a very different position, because a dwarf male would be blocked by another dwarf male that is already in the waiting position. Yeah, the waiting position is lying adjoined to the female in the shell. And if a second dwarf male is around, then it would not be able to even get into this hopeful waiting position. So the dwarf males do apparently not aggregate to enter nests, even though the fish, when they are feeding, when they are not actively trying to participate in reproduction, they form aggregations, and they swim in shoals or even schools, because they're sort of slightly coordinated in their movements and feeding pattern. When they start reproducing and competing for reproduction, they rather seem to avoid each other, or at least they are not attracted by each other.
Okay, thank you.
Okay, thank you. There's one thing I forgot to say, even though we in the meantime all know each other pretty well. When it's your turn for a question, please, can you quickly say who you are and where you're coming from? It might still make it easy also for people that join the fine, not from the beginning. And the next question would be by Ute Radespiel, please.
Yeah, hi, I'm Ute Radespiel from Hanover. Great talk, thank you very much. I like this intriguing combination of ecological factors and sexual selection in that story. What I was wondering is if whether predation may also be a constraint on some of these different sized male classes. So whether being the very small one may also have imply some predation costs or survival costs that may feed also into the system, at least under natural conditions, not in the lab, of course.
Yeah, absolutely. That is a very important point. And that is the reason why we find this sort of similarity in lifetime reproductive success between different male types in the field. Because, I mean, as we have seen, dwarf males are so successful in fertilizing eggs when they get into the position of entering a shell. One would then expect, well, why are there not many more dwarf males? And we saw that it is only about 5% of the population, of the male population, are dwarf males. The reason for that is mainly that dwarf males do not live very long. I mean, they mature early, yeah. I showed that they have a faster growth rate in the beginning of their life, and then they level off very quickly, and then they remain small. And that means, of course, that they are very highly exposed to predation risk. And that means that dwarf males in the field, I mean, we have age estimates from otolith analysis, and we find that in the field, the dwarf males do not get older than roughly about a year. Usually, apparently, most dwarf males die already at an age of 200 to 300 days. Nest males start reproducing at 800 days of age. So nest males have to survive much longer than dwarf males, even to get any reproductive success. Yeah. So predation is extremely important in that whole story. But that was incorporated in our data set with regard to the survival rates of the different male types, and the probability to reach actually the reproductive stage.
Thank you. Then the next question is by, send it privately to me so not everybody sees it, is by Eduardo, please.
Thank you, Karsten. Thank you, Michael. I'm Eduardo from Yale University in the US. Thanks for a wonderful presentation. It doesn't help not be very jealous about all the different levels and ways in which you can ask questions from your system, a combination of field and the lab. And so related to that, you were explaining how the dwarf males are so much more successful in fertilizing the eggs. And I was wondering, have you been able to also get at the level of the cell biology? Is this just a matter of priority of access to the egg, or is there something about the interaction of proteins or components of the cells of the egg and sperm that are giving the dwarf males an advantage over the other ones?
Well, I think it can be very easily explained by the different roles that the different types of males fulfill in that particular situation of fertilizing the eggs. I mean, you should take into account that the dwarf males are right at the spot. I mean, the genital papilla of the dwarf male is very, very close, within half a centimeter of the clutch of each released egg. Whereas the sperm of nest males has to travel on average about 2.5 centimeters from the shell entrance to the place where the egg is deposited, yeah, because the nest male cannot enter the shell as it is too big. So it takes not only a long time, but also a long distance for the nest male sperm to really reach the egg and to then enter the micropyle on the egg, whereas the dwarf male is right at the spot. And that explains really the different performance of dwarf male sperm in comparison to nest male sperm, because dwarf male sperm should be quick to compete with other sperm that is in the shell. But it does not need to survive very long, yeah, because it reaches the egg very soon. Whereas nest male sperm has to be transported, actually mainly passively, of course. I mean, sperm cannot swim 2.5 centimeters on their own accord. It's passive water movements that brings the sperm to the egg before even able to compete with other sperm on the egg or at the micropyle. So I think that makes a lot of sense that nest male sperm are much more long-lived, up to 4 minutes or longer than dwarf male sperm, but that comes at the cost, of course, of swimming performance. I'm not sure whether this answers your question, but I think it's not so difficult to understand the different types of sperm and how you can explain that dwarf males, if they are successful in getting into the shell, are so efficient in fertilizing eggs, even though they of course have much smaller testes, absolutely speaking, not relative to body size, but absolutely speaking, they have really tiny testes in comparison to the huge testes of nest males. But still, they don't need to release a lot of sperm because the sperm are so close, are released so closely to the eggs. Actually, something that is quite interesting that I did not talk about is how the nest males respond to the fact that the dwarf male is inside the shell and participating in spawning. There are three possibilities. Either they should perhaps not respond at all and just continue their fertilization business. Second possibility will be that they increase the amount of sperm because it's sperm competition, and according to Jeff Parker's sperm competition model, if there is competition with 1 or 2 other competitors, then they should increase their number of sperm released. Or it may be that they reduce the number of sperm because they realize that they have no chance in competition anyway, and they rather save sperm for future fertilizations. And actually what we found is that the nest males do the third strategy. So when there is a dwarf male inside, and they can sense this because they always sniff into the shell entrance, and apparently they can sense that there is a dwarf male inside, they continue spawning, but they halve the sperm number per ejaculation. So they really save sperm for future occasions because apparently they are selected to respond in a way that they give up the competition with the dwarf male.
Thank you.
Thank you. Then the next question is by Kaya Tombak, please.
Hi, I'm Kaya Tombak. I'm a postdoc in Jessica Rothman's nutritional ecology lab at Hunter College, New York. Great talk, thank you so much. I was wondering if the female size relative to its shell size affects its ability to control whether she can, whether and when she lets a dwarf male in. If so, then you would probably expect better size matching between the shell and the female under high dwarf male densities. And that could then affect the sort of female's choices and trade-offs between getting a larger shell than she needs and being able to have more offspring versus, being able to control the dwarf male entry success.
Yeah, very good point. This is actually exactly the case. I mean, if a female is relatively large in comparison to the shell size, she benefits from the ability to block the shell entrance against dwarf males, even though she has a disadvantage in fecundity, because not so many offspring can survive or can be produced in such a small shell. So again, this is a trade-off for the females. And you are absolutely right that by selecting shells of different size in comparison to their own body size, they could have a strong influence on the probability of being affected by the participation of dwarf males in reproduction. But as I said already, I mean, for the females, there's also this problem that nest males run out of sperm after a while. So in the later phases of fertilization, it might be quite good if a dwarf male comes and the female should do better in letting them in. Unfortunately, we don't have data showing, we don't have enough data from dwarf males entering shells at different time points so that we cannot really confirm that hypothesis. But it is likely that that females should be more critical in the early stages of spawning than later. So even this relationship between her own body size and shell volume or shell size is not so clear what would be the optimal solution. And it strongly depends on the situation regarding actually something that I have not mentioned. I should perhaps also mention that the females, of course, compete also among each other for shells. Yeah. So the largest shells are limited. Yeah, I've shown the shell distribution, and there are many shells in these nests that are not really preferred by females because they are too small. So the largest shells are the most attractive shells for females, especially, of course, for large-bodied females. And if a small female enters a large shell, she risks to be expelled by another female, even after spawning. That happens, yeah. And so we did an experiment. Dolores Schutz did an experiment where she had females selecting shells either when there were female competitors in the tank or without female competitors. And when there were no female competitors, they always chose the largest shell. There was no exception. When they had female competitors, they went for the shell that sort of most closely fitted their own body size. So they apparently optimized what you mentioned, the body size, shell size ratio. So apparently they have other trade-offs as well. It's not only the potential participation of dwarf males and the potential oxygen ability to raise offspring. So it's a complicated situation also for females.
Thank you. So we still have time for questions. If you want to type in your question mark, I see that Filippo Aureli has the next questions, and then it's me, and then David.
Okay. I'm Filippo Aureli from Universidad Veracruzana in Mexico. Michael, fantastic talk, very nice research, very nice presentation of all these different things, and of course, a very great species model that you pick to investigate all this stuff. So great stuff, and fascinating about these different type of males. And I'm wondering about the sperm competition idea that the dwarf male uses. It seems that in a way that you mentioned was more a default, since there are not many others around that never do aggression, should be sperm competition. But I would like to understand a little more how you think it will actually work. If I understand correctly, there's only one dwarf male, but a single time in a shell. There's no another one that comes afterwards. This dwarf male will stay the entire time in the shell, and there is also, I don't know how often or many times they reproduce in their own lifespan a single dwarf male. So try to understand how the sperm competition could work, because this seems not work in a way that would be the most obvious way, where you have various females and the males visit the various females and they do sperm competition that way. I'm sure there is a very good explanation. I would like to hear from you.
Mm-hmm. Okay, yeah, if we think of sperm competition in that species, there is sperm competition only between different male tactics. Yeah, there is sperm competition between nest males and dwarf males, and there is sperm competition between sneakers and both dwarf males and nest males. But there's never sperm competition between different dwarf males. Yeah, because as you correctly say, only one dwarf male is in the shell at a time. There's not enough space for more than one. But of course, the dwarf males have very different opportunities to engage in spawning in different nests, the nest males. I mean, as I said, the nest male defends its nest for about 33 days. And that involves not only the phase where they are actively spawning with females, but also the phase where they are just protecting the shells against thieves from other nests and against predators. So the nest male is really limited by the amount of shells it can have and provide to females in their shells, whereas the dwarf males can just go shopping from one nest to the next. And however, it's also time consuming for the dwarf males. What I think I did not mention, perhaps it's important to understand the system, is that spawning, as I said, takes 8 hours on average, that only one egg is deposited at a time, at time intervals between 2.5 and 4 minutes. So with each sperm release of a male, regardless which male type, it can only fertilize one egg, not more. So that's also the reason why the nest males get sperm limited with time, yeah, because they have, we counted the number of times that they release an ejaculate into the shell entrance in an average spawning, and this is several hundred times. It may get up more than 800 times for a single clutch. And then, of course, they can spawn with up to 4 females at a time. Then you can see the limitations of the males. And the dwarf males, they can just focus on the egg release of the female. They only need a very small portion of sperm per ejaculate because they are so close. The genital papilla is so close to the eggs, deposited eggs. So they're in a very different situation with regard to the limitations of caused by sperm competition. I'm not sure whether.
No, if I can follow up, I was thinking about the sperm competition between dwarf males, so within the same class of males. That also is the only way that, or you don't think is working the sperm competition?
No, I don't think there is any sperm competition between ejaculates of dwarf males, yeah, because only one dwarf male is really participating. And they stay actually that is important to know. Once they have entered a shell during any period of spawning, they stay until the end. They do not leave anymore. There is no chance for another dwarf male to get in. Yeah, so they will remain until after 8 hours the spawning has ended, and they then leave.
So related to this, if it's not beneficial for females to spawn with dwarf males, but because of sperm limitation, they might be forced to, why is it not an option for them to have more clutches in shorter intervals? Like they spawn only half today and the other half they spawn in 1 or 2 days when the bogwars male might have enough sperm again.
Yeah, I mean, females invest a lot in brood care. Yeah, they care for the eggs and larvae for, on average, roughly 2 weeks or nearly 2 weeks. If they were spawning eggs in smaller portions, even if they stayed in the same shell and release a few eggs on one day and a few eggs on the next day, and the rest of the eggs 10 days later, then that would prolong their time that they have to stay in the shell and fast. I mean, they cannot feed during that time. Yeah, so the females lose about 1/3 of their body mass during one spawning interval. Yeah, because of fasting on the one hand, and of course also because of their investment in eggs and in brood care behaviors. They show a lot of fanning behavior because otherwise there would not be enough oxygen for the developing offspring. So I think it is necessary for them to concentrate the time of egg release to a relatively short period.
Okay. Then the next question is by David, please.
Hi, I'm a student of Eduardo at Yale. Thanks for your talk. It was super interesting, and this is a super neat study species. I'm mostly interested or curious, I guess I should say, about the role that the sneakers play in, moderating this intrasexual competition between the dwarfs and the nest males. I believe that you said that the sneaker males turn into the nest males, that they're smaller or younger. So I wonder what that transition looks like and whether they can go back and forth from the sneaking tactic to the nest holding tactic.
Mm-hmm. Yeah, I mean the sneaker males are belong to the life history trajectory of nest males. Yeah, so if they survive long enough, then they will defend nests later on. The sneaker tactic, as I have shown, is not a very successful tactic. Yeah, so sneaker males can improve their lifetime reproductive success only slightly by their tactic. However, we need to take into account that not all males survive that long until they can really start to defend the nest. As I said, they are usually more than 2 years old when they start defending a nest. And in the meantime, growing up during that entire long period, they can get a few fertilizations here and there. Yeah, so they may have some reproductive success, even though it is very low. To your question, whether they can switch back and forth between sneaking and holding back and rather growing, this is a possibility, but we do not know whether they really do that. We have information from the field. When you catch males that are roaming about feeding and then also sometimes moving into nests and trying to participate in reproduction, only about 10% of these males of intermediate size, intermediate between female size and nest male size, have actually active testes. So apparently either only 10% of the males ever pursue the sneaking tactic before they become nest males, or each male on average switches on its gonads only for 10% of the time. So these are two possibilities that we cannot, with our data set, we cannot distinguish. but I think in principle it is possible that they can switch on their gonads if, for example, there is a very active colony around where they can participate in spawning. And if that colony has gone, which is actually a dynamic that really happens in the field, then they can reduce their investment in sperm and rather invest in accumulating fat reserves, as I have shown that the sneakers really carry quite a bit of fat to improve their chances to be successful as a nest male later on.
Thank you. I quickly want to jump in with a question before we come to Suleima because of this. You say they can switch on their sperm production. Can this be so quickly in fish? I know in mammals it takes about 2 weeks, which is the reason why in many species mammals might remain reproductively active while females are not, so that because it would take them too long to be able to produce sperm again. Is it different in fish?
Well, of course, it may not go instantaneously, but it has been shown in several fish species experimentally that they can even change the type of sperm they produce within a matter of a few days. So and also the growth of testes and the reduction in size is something that can happen in relatively short time periods. Not hours and perhaps not 1 or 2 days, but I think within a week or so, I think they can really strongly change their investment into sperm production as against accumulating reserves.
Okay, thank you. Then the next question, like I said, is by Suleima, please.
Yes, Suleima Tang Martinez. I'm a retired professor at the University of Missouri, St. Louis, in the US. And first of all, Michael, I have always loved your work, and this was a great talk. Really appreciate it. I have a question. Once the nest males become sperm depleted, how long does it take for them to recover? And also during that recovery period, do they continue guarding, trying to ejaculate? Because it seems that would be an ideal time if the male at all backs off during that recovery period when it doesn't have sperm, for sneakers to take advantage of that and for the males to come in. And you may have said this, I'm not sure, but if that were the case, it also seems that females should be much more receptive to dwarf males during the later part of spawning, when the nest males are more likely to have run out of sperm.
Mm-hmm. Okay, yeah, these are several important and interesting questions, which I'm afraid I can only partly answer. So regarding the spawning, if you think of the length of the spawning period of fertilizing or even depositing a single clutch, and this takes on average 8 hours. That means that this is the majority of a daytime light period. They do sometimes extend spawning over to the dark period during night. But in the tropics, of course, the daylight hours are limited to about 12 hours a day. So one spawning fills the majority of the daylight period that is available to them. So usually the nest males, when they are running into sperm limitation, this is already quite late during the day when they do not accumulate new females. Yeah, so they can easily recover until the next day when they start spawning again. Yeah, whether the sperm limitation causes sneakers and dwarf males to more actively trying to get into the shell during the late stages of the fertilization process. This is very plausible, very likely, I think, but we don't have enough data to really prove that. I should say that, of course, in the field, in the natural situation, it is very difficult to to observe the participation of both sneakers and dwarf males. So our observations there are really limited. Also because of diving times, of course, and things like that. But I think that the really crucial thing is whether females and how females respond to that period where nest males run out of sperm. One quite interesting and intriguing trait in this species that I was not talking about is that nest males start to release sperm right away when a female has entered the shell. Females start depositing eggs only after 1 hour.
Wow.
Sometimes even longer, but on average is after 1 hour. So the nest male deposits sperm every 2 to 4 minutes already during the first hour without any effect on fertilizing eggs. So we think that this is an important information for females about the sperm production and availability of nest males, because a nest male may have spawned recently with another female and then it would not be able to release so many sperm. So perhaps the female has an ability to sense the quantity of sperm that the nest male releases before she even decides to spawn. During the first hour, she may also leave, and search for another nest male. So apparently the nest males really have to invest into sperm production at the time when there's no fertilization to gain, just to convince a female to stay and start releasing eggs.
Wow. And there was the other question as to whether during the time that they're sperm depleted, the males continue to defend the nest and continue trying to ejaculate.
Mm-hmm. Yes, of course. Of course, they continue. I mean, if we had not measured the amount of sperm they release per ejaculate, then we had not known that they are sperm depleted. From outside, you see nothing. I mean, actually, it's not completely true. They reduce also the ejaculation release rate slightly. Yeah, so there is a response also after 5 to 6 hours. They release ejaculates at longer intervals. But apart from that, they defend the shell and the nest irrespectively of sperm depletion, and they still continue in releasing sperm, but ejaculates, but with much lower sperm numbers, as you have seen from the graph.
Thanks.
Thank you.
So dwarf males, when they are successful, they're highly successful. But you said females do not really prefer them, probably because the bourgeois males, when they smell that there are dwarf male offspring in it, they empty the entire shell. So that's very costly for the females. But you also showed or implied that it doesn't matter how many of the offspring are fertilized by the dwarf males. Wouldn't this mean for the females it should be a 1-0 game if the dwarf males are successful for whatever reason? Then, because dwarf males on average are so good, they should produce as many offspring with the dwarf males, because the cost is not higher than if she has only a few. The risk that the bourgeois males kills the entire brood. But if the brood is with dwarf males, the more dwarf males are the farther, the better. For the female?
Yeah, not really, because I mean, from the experiment of Jonas Leuenberger, we know that there seems to be a threshold number of foreign offspring from which onwards the nest male will sense the foreign offspring and try to expel the female. Perhaps you remember that he varied the relative and absolute frequencies of dwarf male and nest male offspring between 10 and 40. Of course, we don't have any numbers in between, unfortunately, so we don't know exactly where the threshold is. But if there are only 10 dwarf male offspring or foreign male offspring sired, then the nest male does not increase its shell manipulation behavior, which finally can expel the female. But if it is 40, then they are apparently responding. So I mean, of course, you could say, well, if it's 40 or 100, it doesn't matter. But I mean, once a dwarf male has entered the shell, the female can not influence the success, the relative success of the two competing males, the nest male and the dwarf male anyway. So it's only a question of letting a dwarf male in or not. And of course, to us, it seems ideal if the female would wait until the nest male is sperm depleted, say after 6 hours or so, and then let the dwarf male in. But then, of course, a dwarf male must turn up in the right time period. And as I've shown that the dwarf males are rare, this is perhaps also unlikely that in that particular situation, when it would be optimal for the female to let the dwarf male in, the dwarf male is around and would like to participate. So it's not so easy for the females really, I think, to find the optimal solution to this trade-off.
Okay, thank you. Then you showed in, and then David can ask again. You showed in one slide the time budgets between bourgeois males and dwarf males, where the dwarf males, like expected, don't show any aggression. What I didn't understand, though, you mentioned something, is the dwarf males were eating a lot and the bourgeois males were not eating. And you said something about the period of brooding or tenure. Does it mean the bourgeois males are only at a nest for a specific time, or what was this relating to? I mean, obviously they must eat at some stage.
Yes, of course they must eat at some stage, but they do not eat during the entire nesting period.
So what is this? Is this
33 days on average, so over a month. So during the entire period of defending a nest and releasing so many ejaculates and investing so much in defense of the nest, in courtship and in collection of shells and everything, they do not eat a bite. They fast.
But they stay at the territory all the time, also
all
outside
the
of the...
All the time. They don't leave it for a second, because if they leave it, it will immediately be taken over by a competitor.
So that means there's also synchronized breeding between the females of the nest.
Yes, right. Yes, of course. Synchronization is partly due to the fact that, of course, a male is only actively courting for some period. But this is also triggered by the moon period. So the major reproductive period, the spawning period, is around full moon. And that has a good reason, because at full moon, I mean, I should start differently. The development of eggs and larvae takes 2 weeks. Yeah. So after 2 weeks, the larvae, the little young fish, leave the shell, and they leave the shell at night because there is less predation risk at night. And of course, there is even less predation risk at night if the night is dark. So they leave the shell preferentially around new moon, which means that you should spawn at full moon to have offspring developing 14 days within the shell that leave the shell at the optimal time at new moon. So there is a strong synchronization of spawning. Of course, the spawning continues, and there is also some spawning during new moon, but the moon cycle has a strong influence on the frequency of spawnings.
Okay, thank you. That made it clear to me what I missed. Then another question by David, please.
Hi. So I think you said that the females have some form of like brood care. So I'm curious about the timeline of the brood care and the egg hatching, and both the females and the dwarf males leaving the shell, and how, is there any opportunity for like defense of these broods, either by the females or the dwarf males? And then also, how exactly do the nest males like dispose of these snail shells that they've rejected? Do they just kind of chuck them and the eggs go flying and get eaten, or are the eggs like somehow affixed to the shell? How exactly does all that work?
Yeah, okay. So the spawning, as I said, takes about 8 hours. The dwarf males leave when the spawning is finished. if a dwarf male has entered the shell, which occurs rarely, but if it happens, then they leave after the spawning has ended, and they do that usually during the night, probably because they are safer. I should also point out that a dwarf male can be easily eaten by a nest male. Yeah, we have seen dwarf males being killed by nest males. We have never seen that they are cannibalized, but in theory they could. Yeah, so the size difference is so large, and nest males are, if they get hold of a dwarf male, they are really extremely aggressive against them. So the dwarf males cannot engage in any brood care anymore because they are leaving after the spawning has ended. And of course, that is also their great benefit. They don't need to invest in brood care because that is done by the female and by the nest male. And they could do little anyway, one should say also. So the brood care of the female involves mainly fanning. Yeah, so the female always ventilates with one pectoral fin of fresh water into the shell for 14 days, for 2 weeks, before the young have grown up to a stage where they can live and survive independently. The eggs take about 3 days to hatch. During the period when it is still eggs, apparently the nest males cannot sense if offspring have been sired by foreign males. So the eggs are safe from the male manipulations. Once the larvae have hatched, as I showed in the early stages, the first 3 to 5 days after larval hatching, the nest males can sense whether there is a large number of foreign offspring sired by foreign males. And then they start manipulating the shells. I only, because of time constraints, I only explained very shortly one way how they can expel a female, and that is by taking it up and turning around the shell and throwing it about, so to speak, which is of course not very good for females, because we know from fish that if you keep a fish upside down instead of upright, that is physiologically stressful. Fish that are kept upside down are really damaged after a while. So the female, what does a female do? I mean, why do the females leave the shell when the male throws the shell about? This is because the female has to get out of the shell and turn and enter the shell with the right side up again, with their backside up. And that is the possibility of the nest male, if he is around, to grab the female and expel her from the territory. And that is actually what they do. I have a nice video sequence where I show that, which shows, I cannot show it now because I don't have it available on this computer, but it shows that the nest male takes up a female, brings it to the edge of its territory, releases it, the female gets back into the shell as quickly as she can. The nest male shovels sand into the shell, which is another way to expel females. They just cover the shell completely with sand. Within a minute, the shell can be completely covered, which would mean suffocation of the female. And actually, we found this tactic of the nest male by finding dead females under heaps of sand with a brood. And after the fourth incident, we really were interested in how that happens and what is the reason behind that. That was in the early stages of our studies. And we found it was always in a case where a nest male had stolen a shell from another nest male. And of course, he was not interested in the offspring of and the female, because the female had completed spawning with the other nest male, but they were interested in the shell. So they covered the shell with sand to expel the female, and once the female is expelled, then they can use the shell for attracting additional females. A very mean, tactic of the males. But anyway, I mean, the female, either by covering, if there's enough sand, by covering the shell with sand, or by this manipulation, they have to leave the shell to get in either without sand or in the upright position, and that is the possibility of the male to interfere and grab the female and just expel her aggressively from the territory. I'm not sure whether this answers your questions. I just was a little bit storytelling about this interaction between males and females, which is really fierce.
Yeah, that's very interesting. Thank you.
Then another question by Eduardo, please.
Thank you. One is a follow-up to one of David's questions. I imagine, David, that maybe you were thinking of the floaters in owl monkeys. I mean, the closest we can get to some of the morphs here are the sneakers. So Michael, that very nicely done graph summarizing the different options for these animals to be either on simultaneous or fixed. I think you did have a reference there. Do you know the schematic that I'm talking about? And do you remember where is it that I can readily find it?
Yeah, this is in a book that we edited on alternative reproductive tactics in 2008 for Cambridge University Press, and there we had an introductory chapter that just explained the major questions and the, say, general biology of alternative reproductive tactics, and there we had this graph.
Okay. And a follow-up question, which takes us away from maybe the intellectual sphere. For those of us who've never looked at animals underwater, I was wondering if you could comment a little bit. I just have no clue how you're doing it and how it's changed. It's changing for us. I mean, there have been new technologies that we're bringing to the field, trying to get some data from primates in the canopy. I was just curious, as a natural historian, how have things changed when you do those behavioral observations from the 90s to now? How are you doing it?
Yeah, I mean, of course, to study animals underwater, you have to dive, and that has not changed. Okay. And the diving gear, the diving equipment has not changed so dramatically during the last 20 or 30 years that there is a lot of difference. Of course, the possibilities to obtain data without your presence by putting up a GoPro next to a nest or something like that, that has greatly increased. But already in the early times when we started to work on calyptros, actually more than 25 years ago now, we were using cameras in the field. We had the cameras connected to a tape recorder on the coast, which limited our availability of nests that were close enough to allow that connection. Because when you connect cameras with very long cables, then the loss of information is too big that you cannot cross a certain distance. So we had these kinds of technical problems, but in principle, we were able to take full-time behavioral recordings by that technology already 25 years ago. So, I mean, there are advantages, things are getting easier, but in principle, I think during that period that we were studying this fish, the technical advances were not so dramatic that we can do really completely different things.
How long is a dive? So we go out, we talk about observer fatigue. So in the forest, so you go down and do you stay an hour, 2 hours? I suspect that I'm not the only one who has absolutely no idea of the logistics of this. So do you try to standardize that? How do you do it?
Yeah. No, of course, that depends on the depth in which you are working. So fortunately, these cichlids that we are studying, also the corporately breeding cichlids I was not talking about today, usually dwell in relatively shallow water, even though it is in the range of, say, the first 15 meters or so. So our calyptros work was mainly done in depths between 6 and 12 meters. And that is a depth range where you can stay quite long times. I mean, we stay up to 5 hours at a dive, which is a bit demanding, but that is at least possible without any decompression problems. So it really depends on the depth and on the work you do. I mean, if you just do observations, which of course as a behavioral ecologist, we spend a lot of time observing without any activity, then it can stay longer because you have air that lasts longer. Yeah. So we have usually we have 14-liter tanks. We have double tanks supplying us with 14 liter of compressed air. And with that, we can stay in this depth range that I mentioned up to 4 or 5 hours without much problems. If we are not very active, when we do experimental work and we have to arrange things and to move a lot, then of course the time is much more limited. And we usually have between 2 and 3 dives a day. So diving times are quite extensive over the day. And on average, I would say we spend, really depending on our activity and the kind of experimental setup that we use, I would say on average perhaps 4 to 5 hours underwater a day.
Thank you.
So thank you, Michael, for a wonderful talk about your amazing work on the cichlids, especially Calypterus. Thank you for everyone for a very interesting and fascinating discussion. For me, this was a very good example how theory-driven research is really extremely interesting and fascinating, but also that it can only be done if one has a deep understanding of the ecology and how this influences the life history of the species one is studying, so that this has to go hand in hand. And with this, I'm stopping the live screen on YouTube. Thanking everyone again.